Now, what is going to happen next when this kind of complex is formed on the surface of the macrophage? Now, this sensing via TLR4, it starts synthesis of the inflammatory cytokines, as I told you. You know, this is the complex that you see here. This is the TIR domain of the toll-like receptor 4. And here you see in pink here, this is an adapter protein. And this adapter protein is called MYD88. Okay? Why it is called an adapter protein? Because this is going to combine two signaling molecules together. The TIR domain with another signaling molecule which is known as IRAK4. Okay? What is IRAK4? Interleukin 1 receptor associated kinase. It means it's a kinase. It's an enzyme that would do phosphorylation. Okay? So, when this kind of assembly is formed on the surface of the macrophage, it would bring about changes in the TIR domain, which is on the inner side of the membrane, on the cytoplasmic side. This is going to move. Now I will tell you, that is next one. First we discuss the job of these, what is happening here, the yellow. The TIR domain here, when this assembly is formed, this TIR domain is going to move. Okay? Our conformational changes would take place in the TIR domain. And when some conformational changes takes place in this domain, the yellow one, it would activate this particular protein MYD88, which is also there. And one part of the MYDD protein, when it is bound to the TIR domain, is activated. This domain, it is complementary to IRAK4's, a domain of IRAK4. It can just, bind with that particular peptide. And this actually is an enzyme, which I told you is interleukin 1 receptor associated kinase. It means that this kinase enzyme has now been, activated as a result of this assembly here on the surface of the macrophage with the help of an adapter protein which is MYD88. यहाँ तक समझ आई? Now, what would happen next? You can see that there are two MYDD peptides which are associated with two TIR domains. These are homodimers of TLR4 that you see here. Not all the toll-like receptors are forming homodimers. Only type 1 toll-like receptors form homodimers. The rest of them, they form heterodimers. So many of them form heterodimers, but some of them form homodimers. Homodimers means these are same receptors, and they would just, when they are activated, they would just, come together. And the receptors work like that, Their activation needs, such kind of binding of the dimers with each other, whether these are homodimers or heterodimers. When they bind or when they are expressed together on the surface and they are attached to antigens, then they are activated in that way. No receptor works alone. That one receptor, it would just, bind to a single piece of antigen and it would be activated. So this toll-like receptor 4 also works in the same way, just like other receptors do. Now, what happens that when IRAK4 is activated, this is a kinase, now it's going to, phosphorylate the next peptide in this, row, which is known as TREF6. So TREF6 also is an adapter protein in here. And the TREF6 is now going to be, phosphorylated, and there would be a series of events, phosphorylation and dephosphorylation of different peptides in this pathway, ultimately leading to the phosphorylation of IKK, or inhibitor of kappa beta kinase. Okay? IKK are inhibitor of kappa beta kinase. Now what is IAKK? IKK phosphorylates IKB, inducing its degradation and the passage of necrosis factor kappa beta to the nucleus. What is that actually? This is going to do what? Activate the tumor necrosis factor kappa beta, okay, by phosphorylation. Is going to phosphorylate what? NFKB. And normally it is in this form inhibited by IKB in cytoplasm. Normally, if you look here, this is NFKB, or necrosis factor kappa beta, and this is IKB, inhibitor of kappa beta kinase. So normally this necrosis factor kappa beta, what is this factor? This is actually a transcription factor. But this transcription factor, which already is preformed and it is there in the cytoplasm, it is not in active form, it is in inactive form, because this factor, inhibitor of KB kinase, is bound to it. As you see here, this thing is bound to the blue one, NFKB. Now this assembly should be disrupted and it should be broken so that NFKB is released. NFKB is now not in free form, it is not active yet. But if you just remove this part, the orange one, it would be activated. So what is the orange one? Inhibitor of kappa beta kinase. IKK now is going to phosphorylate this, come here, and this phosphorylation would release necrosis factor kappa beta, and this factor also would be translocated into the nucleus. It's a transcription factor, and the job of transcription factor is to go there inside the nucleus and start transcription of particular genes. There would be transcription factor binding sites for NFKB and different cytokine genes, for instance, other macromolecules or adhesion molecules required during the process of inflammation. Okay, so whatever different gene products are needed for the process of inflammation, all those genes would be transcribed with the help of these transcription factors. In your molecular biology course, you would have gone through cis-acting elements or the role of different transcription factors and how specific and general transcription factors are going to start the transcription of, or enhance the transcription of different genes. Every cell has the same set of genes except the germ cells. Okay, all other body cells, they have the same genome, these 3 billion base pairs, and all of them, they have the same set of genes. But in one cell type, you would see that it's morphologically different from another cell, like the liver cell is different from the kidney cell. The function of the liver cell is different from the kidney cell. The kidney cell is different from a muscle cell. Okay? The muscle cell is different from any other type of cell that you can see. Okay? So how is that morphological and other differences, how are these differences actually maintained inside your bodies? It is carried out with the help of these different transcription factors that are available in one tissue. Specific transcription of factors for liver tissue, for instance, or for kidneys, or for muscles, or heart cells, or any other body type. These transcription factors are not available to other types of cells in other organs, or if they are available, they would be available at certain particular time during the developmental process, or during whenever these transcription factors are needed. And these transcription factors are responsible for differential gene expression. Why these cells are making different products? They are making different, they are doing different jobs. They have different morphologies. They don't look alike, So all these are because of differential gene expression. And that differential gene expression needs different types of transcription factors to be available to these cells in the microenvironment of cells in different tissues. Okay? So that's why in macrophages, what are the macrophages going to do? The macrophages are meant to destroy the pathogens and start this process of inflammation. Okay? Apart from that, what are other jobs of the macrophages that you know? Other jobs of macrophages include to remove debris from the tissues. If there are dead cells, for instance, the cells are dying, apoptosis is taking place, they are going to engulf those pieces of different cells also from the tissues. So the debris that are formed there inside your tissues, normally when there is no infection, no pathogen, the macrophage are busy in doing that job. They are going to just remove the debris out of the system. When there is a pathogen, then the macrophages have got these two things to do. One thing is to just phagocytose and degrade it, and if required, lead to this process of inflammation. To transcribe these genes with the help of these factors and start this process of inflammation. So this is what I already talked about, that NFKB beta induces transcription of cytokine genes, and the inflammatory cytokines are synthesized in the cytoplasm and secreted via the endoplasmic reticulum. Okay? Whatever is synthesized inside the cell is just not simply poured into the cytoplasm of the cell. It has to be contained in some kind of membrane. So everything that is secreted there is kept inside these membranous vesicles, and then they are secreted outside of the cell to do different jobs. Okay? So this is a genetic deficiency of IKK, you know. IKK deficiency can cause developmental problems as well as immunodeficiency. Okay? For instance, this figure shows you this kid with, conical structure of the teeth. This is an X-linked hypohidrotic ectodermal dysplasia. This condition is called. The genetic disease, also called NEMO deficiency. What is NEMO? NF-kappa beta essential modulator deficiency. We call it NEMO. NEMO genes are on X chromosomes, actually. Okay? So this condition is, you say, more common in males as compared to females, because the genes for NEMO are located on X chromosomes. Okay? And the reason is very simple. The male have one X, and female have two. Okay? So if the condition has to develop in females, then both of the copies of this particular gene should be defective. In male, if there is no other X, that's why if one defective copy is there, the male would, develop this condition. So this is not a condition that only affects the teeth. Also, your skin and other, tissues can be affected as a result of this condition. So why it actually is happening? This disease is caused by impaired NF-kappa beta activation as a result of the lack of a functional gamma subunit of the kinase IKK. Okay? The kinase inhibitor, kinase that we talked about in the last slide, which is going to release NF-kappa beta. Okay? So if there is some problem with IKK, that can lead to this particular condition. So NF-kappa beta activation as a result of the lack of functional gamma subunit of the kinase IKK, which normally activates NF-kappa beta. But if it is defective by itself, it would not be able to activate NF-kappa beta. And the result, what would be the result? It would be a kind of immunodeficiency, And among the unusual physical features of these patients are conical or missing teeth, as I show you, but it's not only limited to the development of these conical teeth. Other skin conditions can also result due to the deficiency of this particular thing. So it means that TLR4 based sensing are sensing of the pathogens, and the resultant inflammatory response which should develop as a result of that recognition would not work in patients suffering from NEMO. Okay? So that response would not be activated there in macrophages in all the people who have, some mutations in IKK. Okay? So what do you guess? What would happen to that individual? Immunodeficiency. This individual is going to be very prone to gram-negative bacterial infections. And these gram-negative bacterial infections can be life-threatening in case of people who are suffering from NEMO. Okay? Because the process for the elimination of the gram-negative bacteria begins here inside the macrophages, and this is an essential element of that. Just at the end of the cascade of phosphorylation pathway, this has to release the NF-kappa beta, and defective IKK cannot do that. Okay? Thus, okay. Okay, so we just can discuss the different TLR4. If you look at this table, it would make you a little bit scared. But this is very simple. I'm just going to tell you about 1, 2, 3, and 4. These are the different, types of toll-like receptors which have different evolutionary groups, अगर कर सकते हैं तो बड़ा अच्छा है. Okay? And the pathogens that they recognize, different kinds of bacteria and parasites, as you see there, RNA viruses and DNA viruses, etc. All these are recognized with the help of different toll-like receptors. So we would discuss about these 4 lineages, and what are the lineages that cause, make different homodimers or heterodimers, etc. in the next lecture. Okay?